Hello, and welcome to this video on Tiny Earth. Tiny Earth is a course-based undergraduate research experience, an international network, and an antibiotic discovery pipeline. Tiny Earth engages students in the search for new antibiotics, addressing the global challenge of antibiotic resistance. In this innovative program, students, called Tiny Earthlings, discover antibiotics from soil bacteria in their own communities. Created by Dr. Joe Handelsman of the University of Wisconsin-Madison, Tiny Earth inspires future scientists while tackling a critical health issue. Today, we will go over the purpose and process of this research initiative. To start, we need to understand the concept of antibiotic resistance. As the name suggests, antibiotic resistance occurs when bacteria evolve and develop mechanisms to combat the effects of antibiotics. But why is this so important? Well, when humans contract bacterial infections, the first treatment is often antibiotics, which typically prevent the spread and growth of harmful bacteria. However, when bacteria become resistant to these antibiotics, the medications that once effectively treated infections no longer work. This means that common infections can become harder to treat, leading to prolonged illnesses, higher medical costs, and an increased risk of complications or death, posing a significant threat to public health. You might be surprised to learn that soil bacteria are one of the main sources of antibiotics. Many of the antibiotics that we use today, like penicillin and streptomycin, were originally discovered in soil. Soil microorganisms naturally produce antibiotics to compete with other microbes in the environment. By studying soil bacteria, scientists can find new antibiotics to fight resistant infections, which is why projects like Tiny Earth focus on soil as a rich source for potential antibiotic discovery. So how exactly is this done? The first step is to collect soil samples from diverse locations to ensure a variety of factors influence the types of bacteria that grow in the soil. Factors such as temperature, moisture levels, and the type of vegetation present in the area significantly impact microbial diversity, which is essential for discovering novel antibiotic-producing bacteria. Next, the collected soil samples are diluted and plated on agar, allowing the bacteria to grow. This incubation period facilitates the observation of individual bacterial colonies, which will later be tested for their antibiotic properties. Once the colonies are isolated on each dilution plate, each isolate is replated into its own patch through a process known as pick and patch. This method ensures that the bacteria are effectively separated from one another. The escape pathogens are a group of bacteria known for causing hospital-acquired infections and exhibiting high levels of antibiotic resistance. Safe relatives of these pathogens are non-pathogenic or less harmful bacteria that are genetically related but do not cause disease in humans. These safe relatives are commonly used in research and education as models to study the behavior and characteristics of escape pathogens without posing risk to researchers or students. The safe relatives are utilized to test for antibiotic production from the isolated soil bacteria. This involves streaking a lawn of the safe relatives on an agar plate, followed by patching each individual isolate onto that plate. The new plates are then incubated for about a week at room temperature. After bacterial growth, the plates are examined for signs of antibiotic production. If a bacterium produces antibiotics, it will create clear areas known as zones of inhibition around its growth. These zones indicate regions on the agar where the growth of escape safe relative bacteria has been halted or slowed, signaling the presence of antibiotic activity. To characterize the antibiotic-producing isolates, the antibiotic-producing bacteria are regrown to obtain sufficient material for genetic analysis. Once a good amount of bacteria is acquired, their DNA is extracted to identify the bacteria responsible for antibiotic production. This is achieved through a process called PCR, or polymerase chain reaction, which amplifies specific DNA sequences, essentially making millions of copies of the target DNA to facilitate analysis. After amplification, the DNA is sent for sequencing to determine its exact genetic code. The sequences are then compared using BLAST, Basic Local Alignment Search Tool, against known bacterial genomes in a database. BLAST helps identify similarities between the bacterial DNA and other species, allowing researchers to identify the bacteria and assess whether the genes responsible for antibiotic production are novel or already known. This process is crucial for discovering new antibiotics and enhancing an understanding of how existing ones work. ultimately contributing to the fight against antibiotic resistance. The research can be expanded to include the chemical extraction of antibiotics from the identified bacterial isolates. The extracted compounds can then be analyzed at Chemistry Hub to identify promising new antibiotics. Thank you for watching this video about the purpose and process of Tiny Earth. We hope you enjoyed.